Nitrogenous base plus sugar form nucleoside. When a phosphate is attached with a nucleoside, it forms nucleotide. One nucleotide monomer binds with next nucleotide monomer by 3,5-phosphodiester bond and form polymer of nucleotide. Polymer of nucleotide is called nucleic acid. There are two types of nucleic acid, DNA and RNA. Both nucleic acids are composed of sugar, nitrogenous base, and phosphate. The basic differences between DNA and RNA are: DNA is double-stranded while RNA is single-stranded. DNA contains deoxyribose sugar, RNA contains ribose sugar. Bases of DNA are adenine, guanine, cytosine, and thymine, while the bases of RNA are adenine, guanine, cytosine, and uracil. Now I will discuss about DNA. This is single-stranded DNA. This type of single-stranded DNA is found in single-stranded DNA virus like parvovirus B19. One single-stranded DNA binds with another single-stranded DNA by hydrogen bond and form double-stranded DNA, where adenine binds with thymine by double hydrogen bond and cytosine with guanine by triple hydrogen bond. Adenine always bind with thymine, cytosine always bind with guanine. Adenine and thymine are complementary base, cytosine and guanine are complementary base. This is the straighted shape of double-stranded DNA, but in nature, DNA does not exist like this. The shape of DNA or how the DNA exists, it was stated by Watson and Crick. According to Watson and Crick, DNA looks like double helix. Double helix resembles twisted ladder where the rails of the ladder are made of sugar and phosphate and the steps are made of nitrogenous base adenine, guanine, cytosine and thymine. Length of one rep is 34 angstrom or 3.4 nanometer. One rep contains 10 base pairs and the diameter of rep is 20 angstrom or 2 nanometer. Free 3' hydroxyl end is called 3' end. Free phosphate end is called 5' end. In DNA, we know there are two strands. There are 64 genetic codes where 3 are stop codon and rest 61 codes for 20 amino acid. The strand that contains these genetic codes is called coding strand. The direction or polarity of coding strand is 5' to 3'. It means coding strand runs towards 5' to 3'. In case of vertically arranged DNA, the DNA is read from above downwards. So here is 5', here is 3'. In this DNA, this is coding strand. But in this DNA, these strands runs towards 3' to 5' direction and this runs towards 5' to 3' direction. So this is the coding strand. So in this DNA, this is coding strand. In this DNA, this is coding strand. Both strands are antiparallel. How to read a horizontally arranged DNA? It is read from left to right. In this DNA, this is coding strand. In this DNA, this is coding strand. Coding strand also called sense strand. The another strand that runs towards 3' to 5' direction is called template strand or antisense strand.